this is our video experiment for practical 2 and 3, which is Spectrophotometry 2, Sugar Reducing Analysis, and Physical Properties of Lipids and Fatty Acids by Group 7, Subgroup 4.
Hello, my name is Moni Pirit. My number matrix is 106045.
Hello, my name is Roslana binti Rafael. My number matrix 106168.
Hello, my name is Han Huixin, number matrix 104280.
Hi, my name is India Sejaling. My matrix number is 103748.
Hello, my name is Siti Nur Ayumi binti Badauddin. My matrix number is 106395.
The main objectives for Practical 2A is to introduce students to another application of spectrophotometry, especially in preparing a glucose standard curve using known glucose concentrations. For Practical 2B, the objective is to guide students in using the DNS method to quantify the sugar concentration in a given sample by referring to the standard curve prepared earlier. Before we begin the experimental procedures, let's take a look at the materials and the functions for Practical 2A and Practical 2B. First, the DNS region is used to detect and measure reducing sugars by producing a color change. Next, Herschel salt is for stabilize the color complex, allowing for clear and accurate absorbance readings. Micropipettes and tips are used to accurately measure and transfer small volumes of liquid. We also use test tubes to hold the reaction mixtures, while the test tube rack can keep them stable throughout the experiment. Then, glass beakers are used to hold the test tubes during heating, and the hot plate provides the heat for the boiling step. Cuvettes are used to contain the final solution for measurement. Distilled water serves as a blank in spectrophotometer to calibrate the spectrophotometer standard glucose solution. Finally, the spectrophotometer measures absorbance at 5075 nanometer to determine the concentration of glucose. Material specific for Practical 2A. Standard glucose solutions used to prepare the glucose standard curve. Material specific for Practical 2B. Sample solution tested to determine its glucose content using the DNS method and the standard curve from Practical 2A. Let's move on to the procedures. For Practical A, we will prepare a glucose standard curve using known concentrations of glucose solutions. First, prepare 6 clean test tubes and label them from number 1 to 6. According to the table provided, pipette standard glucose solution and distilled water into each test tube. Then, add 1 ml of DNS region into each test tubes and gently mix the contents using vortex mixer. Next, place all the test tubes in boiling water and heat them for 7 minutes. After heating, remove the test tubes and cool down to room temperature. Once cooled, add 1 ml of Rochelle salt solution to each tube. Mix the solutions well using a vortex mixer. Then, transfer the contents into cuvettes and measure the optical density at a wavelength of 1575 nanometer using cuvette number 1 as the blank. These readings will be used to plot the glucose standard curve. For Practical 2B, we will determine the glucose concentration in unknown samples by referring to the glucose standard curve prepared earlier. The procedure is almost the same as in Practical 2A, but this time we use unknown samples. In these practicals, we successfully analyze reducing sugars using the DNS method. In Practical 2A, we create a glucose standard curve and observed a clear increase in absorbance at 1575 nanometer with increasing glucose concentration. The resulting linear equation y equal 1.3123x plus 0.1212 with an R-square value of 0.9591 indicated a strong correlation. A visible color change from pale yellow to dark brown confirmed the reaction between DNS and glucose. In Practical 2B, we applied this standard curve to determine the glucose concentrations in diluted liquid samples. The absorbance readings range accordingly, with higher concentrations giving stronger absorbance values, while the blank sample recorded 0. Glucose levels were estimated between 3.30 and 10140 grams per liter, aligning with our expectations. Overall, both experiments meet their objectives by demonstrating that the DNS method is effective for both constructing a standard curve and determining unknown glucose concentrations accurately. The main objective for Practical 3 is to investigate the physical properties of lipids and fatty acids, particularly their solubility in polar and non-polar solvents. Now, let's start with the materials and functions for Practical 3. First, we use a microcentrifuge to spin samples and spread components based on density. Next, a dropper was used to transfer small amounts of liquid accurately. Distilled water served as a control to compare solubility. Toluene was used as a non-polar solvent to test lipid solubility. Finally, we used 3 selected lipid-based food samples, which were the main substance tested in this part of experiment. Experimental procedures for Practical 3. We will test the solubility of lipids in water and toluene. Begin by preparing 2 sets of clean microcentrifuge tubes. Into each tube, place 5 drops or a few small bits of each sample. Record the appearance and odor of every sample. For set A, add 1 ml of water into each tube. Then, shake the tubes gently and observe whether the lipids dissolve in water. For set B, add 0.5 ml of toluene into each tube and shake gently. Record all the observations carefully for both sets. In Practical 3, we examine the physical properties of shortening, ghee, and olive oil. All 3 samples were insoluble in water but dissolved in toluene, a non-polar solvent. When mixed with water, the lipids either floated or separated, while in toluene, they form clear or uniform mixtures. These observations support the principle that lipids are non-polar substances and follow the rule of like dissolves like. Overall, the results match the experiment's objectives and clearly demonstrated typical lipid solubility behavior.
Throughout this practical, we gained valuable hands-on experience in constructing a glucose standard curve using the DNS method. We also learned how to operate a spectrophotometer and accurately interpret absorbance value. In addition, we observed how lipids behave differently in polar and non-polar solvents, helping us to understand their solubility properties. We faced some challenges during the experiment. It was difficult to prepare small value considerably, which affected the absorbance result and the accuracy of our readings. We also find that some lipid samples did not dissolve clearly, making it hard to judge their solubility. On top of that, we had limited time to repeat certain steps or verify results through replication. To improve future experiments, we suggest practicing pipetting beforehand and using property calibrate micropipettes. For lipids test, applying gentle heat could help dissolve the sample better. And finally, we believe allocating more time or simplified sentence step will give us the chance to repeat measurement and changes our overall data reliability.